So hello guys, good day, and we are down to the discussion of resolution and detection of nucleic acids. So for the outline, we will be talking about electrophoresis. What is electrophoresis? So a brief definition would be that it refers to the movement of molecules by an electric current. So what molecules are moving, what fragments we are trying to refer to our DNA or RNA fragments. And then electrophoresis will always entail gel systems. So we have the traditional one, the classical one, the Agarose gel electrophoresis. And if we are trying to resolve very small DNA fragments or single-stranded DNA, so we have polyacrylamide gels. And for if we are looking for pharmaceuticals or anions, we have capillary electrophoresis. But it can also be used, capillary electrophoresis can also be used for nucleic acid resolution and detection. So in this discussion, you will also be learning what are the things that compose the electrophoresis equipment. So basically, we will have the gel box or our gel bath. We have the electrodes and we have the casting trays. We have the combs. And of course, our gels in the form of agarose and polyacrylamide gels. And gel loading. So what are the things that we really must not forget if we are loading our sample to the gel? And detection systems, because if you will ask me, without our dyes, we cannot observe our bands. Like we cannot observe them properly. So we need to add uh, RNA specific dyes or DNA specific dyes for us to be able to detect our nucleic acids if ever they are present in our samples. And then for the objectives, you need to learn the principle and performance of electrophoresis. So why we need to, because the first topic was about nucleic acid extraction, and after extracting, for us to know that if we really have the DNA in question or RNA in question, so we need to resolve them and by using electrophoresis. And then we need to compare and contrast the agarose and polyacrylamide gel polymers. So agarose, again, this is the classical, this is the native gel electrophoresis, but there are some instances, or there are really instances that we cannot just resolve large fragments or small fragments because page is fit for the determination of very small fragments of DNA and single-stranded DNA. That's for our page or polyacrylamide gel electrophoresis. And then what else? We also have the principle and performance of capillary electrophoresis. So the widest application for that is for the separation of organic chemicals such as pharmaceuticals and carbohydrates for capillary electrophoresis. And then on an overview of buffers and buffer additives. So buffers, they provide protection to the migrating fragments that we have. They protect the samples from sudden or drastic pH changes. So the buffers are very good in that, and buffers carry the electric current. It's not the gel that carries the electric current in our electrophoresis setup, but it is the buffer. And our buffers require buffer additives. Why additives? Because think guys that a very good an ideal sample should be single-stranded. But there are instances that our single-stranded samples or fragments of DNA or RNA tend to reanneal. So to prevent reannealing and to prevent them from refolding, especially for our RNA, we need to add buffer additives. And specifically, they are known as denaturants. And they are next, we also have the general types of equipment used for electrophoresis, and we have pulse field gel electrophoresis if we are trying to detect the presence of genomic-sized DNA or RNAs, very large fragments, ranging from like 50,000 to 250,000 base pairs. So what's fit for that will be our pulse field gel electrophoresis. We cannot use our agarose, our conventional, classical, typical agarose, and our polyacrylamide is no match for our pulse field because this one is fit for the smaller, very small sizes. This one is for the very large mega base pairs of a DNA. And we also have compare and contrast detection systems because after resolution, we need to detect. And for us to be able to detect, we must use nucleic acid specific dyes in the form of ethidium bromide, and we have cyber green, and we can also use silver stain here, like this one. So resolution and detection, they are done in several ways. Like we can use capillary and gel, but the gel format is the most commonly utilized. And electrophoresis, as what I have said earlier, it is the movement of molecules by an electric current, and it can happen in solution, but doing electrophoresis in gel matrices is really an advantage because it can limit the migration and it can really contain our migrating materials or RNAs and DNAs, or nucleic acids. And this one, the ionization and the part of our phosphate group on our DNA polymer, it gives our DNA a negative charge. So under an electric current, our DNA will migrate toward the positive pole or the anode. So always remember, what's the general rule for electrophoresis? Sample application is always at the cathodal end, and our migration of our migrating materials, whether it's a DNA or an RNA, will always be towards the anode. So migration towards the anode, sample application at the cathodal end. Cathode, sample application, migration, anode. Okay, and another general rule is that if the format is horizontal, like this one, it's just lying, that's back. The gel is laying on its back, so that's what we call an agarose gel, horizontal. But if in a presented in a vertical setup, it's known as your polyacrylamide gel electrophoresis. Okay, and always remember, guys, that there is this another rule that DNA fragments will therefore migrate at speeds inversely related to their size. What do you mean by that? That the bigger samples, for example, how can I explain that? If they are huge, they are having like more than, no, forget the base pairs, but they are big and they are, how to say, positively charged. It's hard for them to move towards the sieve because our agarose or our acrylamide gel looks like a sieve. It's like a filter, like this one. And not this one. So this, for example, these globular things here, for example, our DNA or RNA or our protein mixture, if they are huge, they tend to settle near the cathodal end, the topmost part. But the smaller fragments can really sift and move themselves towards the anodal end because our the molecular structure of our page and that of our agarose is like of a sieve or a filter. So common sense will dictate us that if they're bigger, then it's hard for them to move through the spaces, right? That's why the concentration of our agarose is important if we're doing electrophoresis. So before going to the specifics, let's talk about this one. So gel matrices. So what is being offered by our gel matrices? Our gels will provide, they serve as, the gel can serve as a support medium, okay? And how can it serve as a support medium for the analysis of our separated components? They can provide resistance to the movement of molecules under the force of the electric current. They can also prevent diffusion of our migrating fragments, and also they can decrease convection currents. So eliminating all those things, we can surely produce a defined group, a distinct group known as a band. So our result will always be something like this. So we will be having bands. This one, this is a good band. These are good bands on the leftmost column. But the second and the third column, these bands are smeared. So these are not good bands. This is an example. This the second and the third columns are examples showing us that when the purity of our agarose, of our gel, during preparation, contaminants were introduced, there's a tendency that our bands tend to become smeared. So is this acceptable? This is unacceptable. Okay. So what else? Oh, method. See, this is the way how to load it. This is our Agarose gel electrophoresis setup. And the good thing, we have this in our lab. So here, gel matrices again provide support, and we need to have the distinct band formation. And then the good thing, it's simple to prepare the agarose. So let's talk about agarose. So what are agarose gels? So agarose, guys, is not agarose directly. It came from the red seaweed, the red kelps. These red kelps are the sources of our agarose. But uh, out from the red kelps, we have agar, and a more purified form of our agar is agarose. Because we have two components of our agar from the red kelps, our red seaweed. We have agaropectin and agarobiose. If we will remove agaropectin, leaving agarobiose alone, leaving the repeating units of agarobiose alone, that's our agarose. So this one. See, agarose is a polysaccharide polymer extracted from seaweed, and it's a component of agar used in bacterial culture dishes. So agarose is a linear polymer of agarobiose. So again, from the red kelps and the red seaweed, we get agar, and agar, if we will purify it further, we will get agarose. And agarose is specifically the repeating unit of agarobiose. So that's for our agarose gel. So hydrated agarose gels in various concentrations, buffers, and sizes can be purchased. So we have precast gels like ready-made. So we can buy, but it's easy to perform this one or produce this one in the lab. So basically, just like your culture media for bacteriology, that you're using powdered NAs like MHA, we have chocolate agars, right? Powdered agarose is suspended in buffer. It's heated and poured into a mold. So heating, you might wonder why we have a microwave in our molecular lab. The use of that is to it is to melt our powdered agarose once it's suspended in buffer. And always remember that the agarose concentration is determinant of the size of the spaces in the gel. So we can perform or produce or make two concentrations. We have concentrated or agarose with a lower concentration. So which one is for what piece of DNA? So for small pieces of DNA, we have 50 to 500 base pairs. It's best for us to resolve them in more concentrated agarose gels. How concentrated? That has to be 2 to 3%. However, for larger fragments, common sense, it's difficult for them to move if the concentration of our agarose is very concentrated. So we tend to make a lower concentration for our larger fragments ranging from 2,000 to 50,000 base pairs. And remember guys, do not ever create or make 5% or above 5% or below 0.5% agarose concentrations because if we will have below 0.5% concentrations of agarose, the tendency of that is the gel will be very weak, will be fragile. So it's difficult during transferring it towards our gel bath or our gel box, and it might not be able to support fully our migrating fragments. How about if the concentration is too high? A very high concentration means that even the small pieces of DNA can no longer move, they are impeded, they can no longer migrate in a very high concentration of agarose. So again, what are the best concentrations? We have zero point 0.5 to 1 and 2 to 3. So that's it. And then I'll skip. Let's move towards the problem when it comes to the purity of agarose. Nowadays, we don't have much problem on the purity of agarose, especially because they are produced commercially. However, if we tend to make mistakes and put inadvertently or accidentally contaminants to our agarose gel or to our agarose preparation, what will happen? So the phenomenon of electroendosmosis might occur. So this refers to the fragment flow. Our DNA or RNA will flow toward one of the electrodes, and most often it will tend to go to the anode. It will migrate towards, no, not the anode, sorry guys, the cathode, the negative pole. So it tends to contradict the general rule that migration is always towards the anode, right? So you might wonder, oh, why is it that my samples aren't moving and they're just at the cathodal part? Is there something wrong? And you might just say that, maybe that's really their nature. No, it could be that you had prepared an impure agarose gel, right, in the lab. And then after these particles or these fragments, there are also instances that you need to resolve very large pieces of DNA, and resolving them in a simple agarose gel is not efficient. Why? Because they cannot move, they cannot migrate. See, and that's known as limiting mobility. And limiting mobility is reached when our DNA molecule can move only lengthwise through successive pores of a gel. So this is known as reptation, from the Latin word reptare, meaning to crawl like a snake. So have you noticed that snakes will just move like lengthwise, they cannot move crosswise. So that's the same case here. So for those genomic-sized DNA molecules, for those mega-base fragments, for those chromosome-sized DNA fragments, for them to move, we need to apply pulses of current in alternating directions or dimensions to enhance reorientation so that they can move lengthwise and crosswise and they can pass through the sieve of our gels. So that's the case. See, also this process is known as your pulse field gel electrophoresis, and it has four examples. We have Fige, TAFE, Rage, and Chef. So the most common, the simplest approach to this method is our field inversion gel electrophoresis, in which our DNA can move. There is a periodic movement of forward and backward movement as we control the temperature and as we have a switching mechanism. So all these things, all these four approaches are under pulse field gel electrophoresis. And always remember that if we're talking about realignment of the fragments at 180 degree, it refers to our Fige. Okay, so again, current pulses application, and then there is reorientation. That's Fige under pulse field gel. Again, pulse field gel for very large genomic DNA fragments, for genomic size DNA, for chromosome size DNA. And then if you're using pulse field, the large fragments are resolved not only by sifting, but also there is reorientation and time necessary to realign. So there's reorientation and realigning so that there's easy migration for the fragments. And then, however, there's a little warning if we are trying to resolve DNA here, because these large DNA fragments are prone to breakage and shearing. So we need to protect them by putting them or immobilizing them in an agarose plug before cell lysis. And then further treatment of the DNA with restriction enzymes is also performed while the DNA is immobilized in the agarose plug. And then that plug is inserted directly to the agarose gel for electrophoresis. So guys, also remember that, please remember that pulse field is a slow migration process. So it will take about 24 hours as compared to our agarose, which can happen just half a day. Okay, so we have alternating field electrophoresis was the application, again, is for the resolution of, it is used to resolve chromosome sized fragments of DNA, and it's fit for in cases like bacterial typing, especially if you're having studies on epidemiology, so for epidemiologic purposes. And digestion of genomic DNA with restriction enzymes will yield a band pattern specific to each type of organism. So after we're able to resolve them, just compare the bands. And if there's a similarity of the organisms or microorganisms, if we're able to isolate them from various sources. Like for example, guys, a good example of this one will be not for bacterial typing, but for viral typing. Right now, SARS-CoV-2 is best analyzed from wastewater treatment. They're no longer testing populations from humans, but they're testing wastewaters. So wastewaters coming from homes, right, and from communities, and those will surely reflect if there's another variant or another a different, how to say, there's an emerging variant, yeah, that is coming if there's a difference when it comes to their band patterns. So what is the methodology that they're using? They're using pulse-field gel electrophoresis. Okay, so after agarose, we're done with the pulse field. Now we have polyacrylamide gels, so PAGE. So if you wanted to dissolve very small DNA fragments and single-stranded DNA, the best approach for that will be the usage of PAGE. And why polyacrylamide, guys? Poly because we have repeating units of acrylamide, and these are repeating units of acrylamide are joined by a cross-linker in our bis cross-linker, bis-methylene acrylamide. So the repeating unit of polyacrylamide is responsible for giving the gel its porosity. That's why the gel is porous. And you might wonder what gives PAGE its stiffness, gel stiffness. Why is the gel stiff? That's our cross-linker, because the presence of our cross-linker gives a covalent bond, and it gives a branching. See? So it gives structural integrity. However, unlike our agarose, if it will polymerize or if it will harden, it will just polymerize upon cooling. So just like our gulaman, just like our gelatin at home, we'll just let it cool by itself, it will harden. But with a PAGE, polyacrylamide gel, this one is like we need catalysts or a catalyst here so that polymerization can proceed. So in the form of our nucleation agents or light activation. So we have two types. We have persulfate plus TMED, and we also have riboflavin plus TMED. So persulfate or ammonium persulfate will produce oxygen-free radicals in the presence of TMED. So what's with the case of those oxygen-free radicals? Those those free radicals will drive the polymerization process of our page. See? So without free radicals, there's no polymerization. But however, we can also use another type of catalyst in the form of light activation, and that's our riboflavin plus TMET. So again, catalyst for the polymerization of our page. Okay, so see, the repeating unit of polyacrylamide is acrylamide, and this introduces branches into the polymer. This is the cross-linker. Okay, so we have acrylamide in combination with its cross-linker. So polyacrylamide was originally used for protein separation, but actually it can also be used for nucleic acid sequencing, mutation analysis, nuclease protection, and we'll be having a separate discussion for this one during P2. So let's proceed. Okay, so for PAGE, polyacrylamide gels you can buy them commercially in preformed gels. And if I'm the supervisor of the lab, I prefer the preform or precast gels because always remember that our how to say, polyacrylamide is a dangerous, see, it's the powdered form of our polyacrylamide is a dangerous neurotoxin, And even if we will combine polyacrylamide with this cross-linker in a solution form, still they are dangerous, though of a lesser degree. So there should be very careful handling if we're dealing with a page in the laboratory. And you will ask me, do we have this? No, we don't have this. We only have the agarose gel electrophoresis. Okay, so guys, here, the composition of our polyacrylamide gel is represented as a total percentage concentration weight per volume of monomer T and that of our monomer C. So monomer T is a combination of the acrylamide with its crosslinker, and monomer T is just the crosslinker alone. The most common ratio utilized in the lab is this ratio, 19:1, 19 parts acrylamide, 1 part of our bis gel, giving us a total value of our T 6% and for our C value 5%. So that's the thing. And going back, unlike agarose gels that polymerize upon cooling, polymerization of polyacrylamide gels requires the use of a catalyst. So again, that catalyst, where is that? Our ammonium persulfate plus timid, or our riboflavin plus timid. So these catalysts, we have two parts, two kinds. We have nucleation agents, APS plus TMED, and light activation. That's our riboflavin plus TMED. Alright, well, guys. Okay, going back, so it's very important if we're preparing the gel, there must be, or there should be an absolute, or bubble should not be present. Why? Because we need to really remove the air, and we should do a de-aeration to because before we will add no, the gel solution is often done before adding it to the nucleation agents. Why? Guys, it's not a good idea that if we have bubbles or air in our page, in our gel, because air, an excess, or if there's too much of oxygen tends to block the polymerization process. Remember, we need free radicals here, not oxygen. See, excess oxygen inhibits the polymerization process, so it will not harden. Or if it will harden and you will still insist on using that, there will surely be a problem on migration. Okay? And think guys, page gels are very thin, like 50 micrometers only. So the preparation of this gel is really difficult. So it's better to buy the preformed or the precast gels. However, the main advantage of our page over agarose is it offers a higher resolution capability for smaller fragments. We cannot resolve small fragments in our agarose gel electrophoresis. We can only resolve 50 to 500 base pairs. Below 50 base pairs, we need to use page. Very small fragments, single-stranded DNA fragments. Okay. And why is it that it offers a higher resolution capability over agarose? Page can detect a variation in as much as 0.1% difference. So like a variation of one base pair in a 1 kb molecule can be detected in page C. And another advantage of page is it's synthetic. So if it's synthetic, differences in the batches obtained will be less. And also we can really adjust the pore sizes based on by changing or altering the T value and the C value. Okay, so that's for our page. And also, aside from the 19.1, you can also utilize the 29.1. So this 30 and this is 20. Okay, guys? For a while, I'm going to charge my laptop. Okay, so let's proceed to capillary electrophoresis. So again, it's for pharmaceuticals and carbs, and for inorganic anions and metal ions. And if you don't have HPLC, the alternative method is capillary electrophoresis, because the advantages include faster analytical runs and lower cost per run. But remember, the equipment for capillary electrophoresis is really costly. It's still expensive as compared to Agarose. And then in this type of electrophoresis, the analyte is resolved in a thin glass fused silica, which is like 30 to 100 cm. So think 1 foot is 34 cm. So this is quite long. And the internal diameter has to be 25 to 100 micrometer. And fused silica, you should remember, associate fused silica with capillary electrophoresis, because fused silica was used because it is the most transparent material that permits a fluorescent light passage. And this silica it has a coating for protection with polyamide, yeah. And it's through the uncoated window where the light is shown on fragments as they pass through the detector. So in this type of electrophoresis, electroosmotic flow is introduced along the length of the capillary, and then it's just the same, guys. So what's the same thing here? This one. Negatively charged molecules are ionized at high pH, whereas positively charged molecules are completely protonated in low pH buffers. So what's the main thing? Large or positively charged, slow migration, negatively charged, and smaller, faster migration towards the anode. Okay, so that's for our capillary electrophoresis. And you will ask me, how much of the sample of the denatured nucleic acid in buffer shall we add or inject to our capillary electrophoresis? It's about 1 to 15 nanoliters, okay? Containing it must have formamide, which is held at a denaturing temperature through the run. So that much sample, 1 to 50. Actually, it's not much. It's NL. And then if we're dealing with nucleic acids, we will use electrokinetic. But some other means of sample injection include hydrostatic and pneumatic injection. So that's not much for our most important thing here. For the resolution of our nucleic acids, the thing with our um, the tube, the capillary small volume, if we will compare it with our agarose gel or polyacrylamide gel, it can dissipate heat more efficiently during the process or during the procedure. So an increased heat dissipation will permit technologies to run at samples at higher charge per unit area. So which means that there is faster migration. So faster migration, there is also faster resolution or runtime. So it can be done faster as compared to the gel electrophoretic techniques. Okay. So for capillary electrophoresis, bacterial typing is for the page, guys. But for this one, it's intended for parentage testing, especially for analyzing short tandem repeat polymorphisms, and also for like things like clonality testing, microsatellite, instability detection, and for the analysis of bone marrow engraftment. So that's for our capillary electrophoresis. Next, let's go to our buffer system. So the purpose or the main reason of having a buffer system is this it is responsible for carrying the current and to protect the samples during electrophoresis, because there's a possibility of pH changes that might happen. So the use of buffers, we can eliminate such problems. So what is a buffer? So it's a solution of a weak acid and its conjugate base. So I think you had a lot of lectures of this one during your CC class, so we will not delve much. So we'll just go directly to the main thing for molecular diagnostics. So here, guys, according to the Henderson-Hasselbalch equation, So for us to change the pH of a buffered solution by 1 point, either the acidic or basic form of the buffer must be brought to a concentration of 1/10 that of the other form. So for example, if you had a pH like pH 8 and you wanted to change it into pH 9, so remember pH 8 is basic, so the acidic form of that has to be brought to a concentration of 1/10 that of the basic form. So just like that. Sorry guys. And then again, we also have the control of the pH of a gel, but the buffer also protects the sample molecules from damage. And furthermore, the current through the gel is carried by buffer ions, preventing severe fluctuations in the pH of the gel. So it's just the same as what I've said. So that's with our buffers. And then the thing that you need to remember with the buffers is if we are dealing with ions having high charge differences, ions with high charge differences like -3, like +3, -2, +2, because these ions, they can really migrate in the gel in a faster way. That faster movement can increase conductivity without increasing buffering capacity. So this will result to much current passing through the gel as well as faster depletion of the buffer. However, for us to, we really have to circumvent these problems. So we need to use trace buffers. We have three types: TBE, TPE, and TAE. It's funny, like some will read this one as TAE. No, it's trace acetate EDTA. This is the one that we'll be using in our lab actually. We also have trace borate and we have the trace phosphate EDTA. So what's with the trace buffers? Trace buffers, they remain partly uncharged at the desired pH and thus they can maintain constant pH without high conductivity, even if we're dealing with ions with high charge differences. So that's the good thing with using trace buffers, because you might ask, oh, what's the use of the buffer in our electrophoretic system? So that's it. They they should they can maintain a constant pH without themselves. They can remain partly uncharged without having high conductivity. So you will be preparing. You will be taught how to prepare our TAE or TAE buffer. Okay, so that's with the buffers. Buffer additives. So what's the main point? The main point here is that, as what I have said, the good or an ideal fragment or sample has to be single-stranded. But if it's not single-stranded, like in instances that they tend to reanneal, in instances that they tend to refold or fold into itself, so we need denaturing agents in the form of formamide or urea, because these agents are able to break hydrogen bonds between complementary strands or within the same strand of RNA or DNA. So denaturing agents, we need to denature again, because it could be that during the process, some of the fragments were able to reanneal. So we need to add buffer additives in the form of denaturing agents like formamide and urea, so that we can prevent the problems on reannealing and refolding. Refolding happens with a single-stranded RNA. Okay, and much for that. We also have the electrophoresis equipment. So we have, if we have gel electrophoresis, we have the horizontal and vertical. Again, if it's horizontal, that's for agarose gel, and if it's vertical, it's for your page or polyacrylamide gel electrophoresis. So in general, agarose gels are run horizontally and page are run vertically. And horizontal gels are run in acrylic containers. Acrylic, so which is an example. This is an acrylic container. See, it's vertical, and you have to in the middle you will be placing the agarose gel slab. It could be a square or rectangular. We also have the electrodes. This is the black part is that the cathodal part, and is the anodal or the reverse. And this one is showing how to load it, how to load the sample in the wells of our gels. Okay, so what comes first? We need to place our gel. slab, and then we will place the buffer. And there's an indicator here. There's a line that will tell us or dictate us at what level we should add much of the buffer, okay? So, okay, we're done with that. So we have the acrylic containers called gel boxes or gel baths. So it has two parts with a platform in the middle. See, two parts with a platform in the middle. Two parts here. And this is the platform in the middle that will hold our gel slab. Okay. And then platinum wires make up the electrodes in the gel. So the wires are connected to a power source by banana clips or connectors. And then in some instances, they call the gel submarine gels. Because why submarine means? Submarine because our agarose gel is below, really submerged below the buffer. For example, below our TAE buffer or TPE or TBE buffer. So if we're talking about submarine gels, it will refer to agarose gel electrophoresis. Okay? So much for that. We also have horizontal agarose gels are cast as square or rectangular. Yeah, we're done with that. And purchased gel boxes come with casting trays. So we need to really prepare the gel before we will place it in our gel box. So we have a casting tray so that our casting tray serves as the mold for our agarose gel. Do not worry, you will be doing this in the lab, so you can really have a hands-on experience on this. So this one, we have the gel, our agarose gel, we have the platinum electrodes, we have the cathode, we also have the anode, the red one. So horizontal, so the submarine gel system. So it refers to our agarose gel electrophoresis. And then see, agarose applied as a dry powder is mixed with a certain percentage weight per volume with our buffer, and it's heated in our, we can use the, how do we call that one? Our, never mind, it's the other one for the microwave, hot plate, sorry. So we have the hot plate or by microwave, and then the microwave is used to dissolve and melt the agarose. And you might ask me, when is the time, what is the temperature that we need to pour it? So if we can reach a temperature of 55 to 65, we can pour it in our casting tray, and then we will not stop with pouring it on our casting tray because we need to place the comb. So the comb will determine, or the comb is inserted into the top of the gel so that we can create holes like this one, So in the form of what it looks like mine, looks like this combs for our page. Okay, for example, this one, or this one is also for our similar for our agarose gel. The shark's tooth comb is this one, somehow different. But the most commonly utilized comb is the upper one. Okay, so we will pour it into a mold, and then we will place the comb so that we can have holes. We can create holes so that we can, and in those holes, we can place our gels. We can load our, not our gels, we can load our samples to the gels. Okay, so see, the size of the teeth in the comb will determine the volume of loaded sample. It's just like 5 to 10 to 15 to 20 UL. Depends on the size of the combs. And so the number of like how many samples you can place or you can load, it is predetermined by the number of the teeth of the combs. Okay. So after the gel has polymerized, the comb is carefully removed and the gel is placed into a gel box and submerged in electrophoresis buffer. So how to do it? First is to melt our powdered agarose and then resuspend in the buffer and then melt it inside the microwave or a hot plate, but it's best to use the microwave. And then after, we need to have it polymerization, but before there's complete polymerization, we need to add the combs to create the hole so that we will have wells for our samples. And then once there is full polymerization, we will remove the comb when it's hard. if it's already hard already, and then we will place it inside the gel box, and then followed by the addition of the buffer. Okay. We also have vertical gel boxes that are separate chambers that are connected by the gel itself, because this one has you might 1, 2, 3. So we also have a buffer for the cathode. We also have a separate buffer for the anode. So that's the rule. Like that's why we have 3 areas or divisions here. And the central part will be occupied by the gel, by the page gel. Okay. So electrodes are attached to the upper and lower buffer chambers to set up the current that will run through the gel. And the gel must be placed before filling the upper chamber with a buffer. So what's the rule, guys? The gel first before the buffer. The gel first before the buffer. Okay? So never the buffer before the gel. Always add the gel first before filling the chamber with the buffer. Okay? Because if we'll do the reverse thing, there's a tendency that there's an overspilling or an overflow of the buffers. And what else, guys? So with our there's a distinct phenomenon that can happen in our page electrophoresis, because gel smiling can happen only not in our agarose but with our page electrophoresis. And this is because on temperature variation. Think, guys, that the outer parts of our gel, of course, is cooler as compared to the central area, right? The central parts, central areas. So even if they are having, if the bands, even if in nature they are similar sized bands, they have the same base pairs. The problem is the bands at the outer areas tend to migrate slower as compared to the ones in the central part. It's because here at the central part, it's more hotter as compared to the parts on the outer lanes. So it tends to, this one, there is a slower migration, but a faster migration in the central part, giving you a gel that tends to appear like a smiling face. Where is that? This one. See, guys, like a gel smiling. Because this one, faster migration, it's hotter, and the outer lanes, it's, how to say, cooler, so slower migration. But actually, they should be on the same line. They should appear on the same level, same line. But here we have gel smiling. According to the scientists, it's a roller coaster effect, but just use the gel smiling for our case, guys. So that's gel smiling, and it happens only in our page electrophoresis, and it is because of temperature variations, okay? So we also have vertical gels are cast between glass plates that are separated by spacers. So for our page, we have glass plates. For our agarose, we have acrylic boxes, okay? And always remember that the spacers determine the thickness of the gel. So the thickness of the gel inside in the central part is 0.5 to 4 mm. So that's the case. Okay, guys. And we also have specialized combs or shark's tooth combs. And usually they are used for sequencing gels. And they are, how to say, placed upside down, teeth up, not in contact with the gel to form a trough or trough on the gel during polymerization, trough or trough. After polymerization is done, the comb is removed and placed tooth side down on top of the gel for loading. So that's the case for the shark tooth comb. So much for that. Let's go to gel loading. So to summarize gel loading, guys, before we will load our samples containing our isolated or extracted nucleic acids or RNAs, we need to place a tracking dye and a density agent. So the tracking dye is for us to monitor the rate of migration of electrophoresis. So there's a dye. in the form of our bromphenol blue and silene cyanol green. And we also have density agents, either fecal, sucrose, or glycerol. Because if you will ask me, Miss, once we will load it, can we really have the guarantee that our samples will not float into the surface? The thing that they're not floating into the surface is because we are supposed to add density agents in the form of sucrose or fecal, right? That's the reason that if you will place them inside the wells, even there's a buffer already, they will not float and mix with the buffer. They will stay inside the wells. And also for us to visualize the rate of migration, we have the tracking dye, okay? Like your bromphenol blue and the silene cyanol green. So that's the thing, okay? And we also have detection system, sorry. So we're done with the resolution part. Now is the time to detect. Now is the time for us to see. So we have nucleic acid specific dyes. We have two parts. No, two examples. We have an intercalating agent. It tends to insert. So an intercalating agent. So we have, for example, we need to search an intercalating agent for a while. Ethidium bromide. Guys, no, ethidium bromide. Sorry. So something's wrong with our net. Don't know why. See, this one's a better example. So an intercalating agent is something like this one. It tends to insert itself in between the, how to say, our base pairs, our nitrogen bases. See, this is, the black one is the ethidium bromide. So it can really incorporate in itself, it can really insert itself in between the nitrogen bases in our DNA helix. So it can lead to sorry, to the distortion of our DNA helix, right? And aside from the distortion, it can give or it can result to mutagenesis, or it can lead to cancer, it can lead to mutations. So we should also be very careful with if we're using ethidium bromide. Don't worry guys, we don't have ethidium bromide in our, we are using a different RNA or DNA specific dye for this case. Okay, so that's the thing with ethidium bromide. It intercalates or stacks between the nitrogen bases in double-stranded DNA. And ethidium bromide has to be excited at what wavelength? UV light at 300 nm, and it will give you an orange visible light at 590 nm. So here, it will emit an orange light when illuminated. So I think this one is the, what is that? Or if it's orange, where is the orange one? I lost it. Somehow I've lost it. Okay. So guys, going back. So it will give you an orange light, or it will emit an orange light when illuminated with a UV light. So this, how can we do UV illumination? Have you noticed that if you enter our molecular lab, there's this black equipment there, and then it reads or it says that it's labeled UV trans illuminator. So that's the thing that we'll be using for our illumination with UV. So after we're done with the resolution, so we will detect our bands by trans illuminating our band or agarose band under UV light. So I think we're using cyber green. I need to check it. Still, this one will still give an orange visible light. Okay. Because guys, after electrophoresis, what you will do with your agarose or acrylamide gel, you need to soak it in a solution of running buffer in like TAE, TBE, or TPE. So you don't have it's wrong for you to like carry it and it's dry and just place it inside a Tupperware or something, a container. No, you should submerge it in a running buffer. So this one in Thai buffer, okay, before or while waiting for your turn to do trans illumination with the UV light. So that's the thing. And then we also have cyber green. Though cyber green is also a nucleic acid specific dye, it has a different principle when it comes to how it uh, joins the DNA or how it attaches itself to the DNA because different or very it's not similar from the how ethidium bromide intercalates or inserts. It does not intercalate between bases, guys. Rather, it sits in the minor groove of the double helix. So this one's the most important line. It sits in the minor groove of the double helix. And in association with DNA or RNA, it also emits light in the orange range. So still orange. So we will not be confused if we are having an exam. And if you will ask me, Miss, between ethidium bromide and cybergreen, which one is commonly used? Because we're trying to avoid the problems on mutation, we're trying to avoid problems on carcinogenesis as they are associated with ethidium bromide, so it's better to use cybergreen. And cybergreen is also the dye of choice. Oh no, for before that one. Cybergreen 1 is for DNA, cybergreen 2 is for RNA, okay? And cybergreen is the preferred dye for real-time PCR methods. Real-time in sense that we can detect the, how to say, we can really observe if we're able, the cycles while we are measuring our DNA or RNA fragments. So the best for RT-PCR, real-time PCR, is cybergreen. And if before, guys, I know you are very well familiar with the silver stain or silver staining in histopathology, but we can also use this in molecular diagnostics. So after electrophoresis, our sample is fixed. So we need to do fixation using acetic acid and methanol. And then after fixation, our gel or agarose gel will be impregnated with ammonical silver with silver diamine solutions, or we can also have silver nitride. So we have two types, our diamine and our nitride in a weakly acid solution. So this will give you an insoluble black silver salt precipitate. So it's black silver. So it's like the how to say that, the iodine thing in your parasitology. The background is like brownish or yellowish and you can see black things. But please do not do overexposure of the gel to our silver stain because it will result in high backgrounds and it can mask the risk. So that's it, guys. Please for our exam we will be I will be including chapters 4 and 5 for this coming January 17 because I have moved the exam so much of the people can be accommodated. And please, 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 I only have one reference for our quizzes, so that's Buckingham. So good luck, God bless for your long quizzes and P1 exams to all subjects. So time to say goodbye.